So we're going to finish up briefly with, oh, I got to hook up. Okay, so we have three choices here, heart, brain, liver. Which one's the most likely to regenerate? Liver. Why? Why does it? What kind of tissue is it? It's gland. Liver is glandular tissue. Yes, it's an organ, but it's made of glandular epithelium, and epithelial tissue regenerates well. Okay? Is there connective tissue in the liver? Yeah, there's some like reticular connective tissue. So it's much reduced compared to the amount of epithelium. If you look at a sectional view of the liver, there's almost no connective tissue there. It's almost all glandular epithelial cells. Okay? So does it make sense to you that your liver regenerates well? Does it regenerate 100%? Nothing ever regenerates 100%. Okay? What is wrong with heart as an answer? What kind of tissue is it? Muscle plus connective. And what do we know about muscle tissue? It doesn't regenerate well. So the connective tissue, of which there's a liberal amount, is going to form scar tissue. And likewise with the brain, what kind of tissues are we looking at? We're looking at nerve tissue. Nerve cells don't regenerate. The neuroglial cells take over the role of connective tissue and form scar tissue. Okay? All right, our second question, why do arteries harden in someone with chronic hypertension? So what was the stimulus that was causing the hardening of the arteries? Calcium was the result, not the cause. What caused the arteries to harden? They're not really going to rupture. They're going to respond in order to prevent rupturing. So what was it? Calcium's after.
It's the pressure.
That's after. It's the pressure. So what caused the arteries to need reinforcement? They were subjected to hypertension, high blood pressure. The high blood pressure stretches the arteries chronically more than they ought to be stretched. And the tissues respond to that stimulus by reinforcing themselves. So first you get fibrosis, and then if the hypertension persists, which it probably will because that didn't solve the problem, did it? Then you get the calcium added. Okay, so make sure you know which comes first, what's the stimulus, what's the response. Are we pretty clear on that? Okay, some of these terms we've already met, but we're just going to kind of fold them all together here. So we know what differentiation is, right? That was one of the terms we learned in the first chapter. Differentiation is the maturation or specialization of cells. They take on special structures and functions and roles in the body that make them different. Okay? But we have a couple of terms associated with differentiation that are abnormalities that I want you to be familiar with. So when you see that a tissue becomes metaplastic, metaplasia, that means it's differentiated abnormally. It's not doing what that tissue is supposed to do. And it is a step towards a tissue potentially becoming cancerous. So when you have cells in a tumor, whether it's a skin tumor, a liver tumor, a lung tumor, I don't care where it is, you have cells that are misbehaving. Do they look normal? No, they're metaplastic, they're abnormal. And when they become anaplastic, anaplasia is they're so hopelessly abnormal, they don't look like any normal cells. So in metaplasia, the cells look pretty normal, they just don't look right. So you might see cells that look like glandular epithelium in the middle of your lung, and they don't belong there. That's metaplasia. Okay? Anaplasia is the cells are so wildly mutated, they don't look like any normal cells. In the early stages of metaplasia, sometimes those cells either reverse course or your body's defenses get rid of them. By the time they're anaplastic, forget it. They're not going to listen to normal signals of the body. They're not going to respond normally. And your best hope for anaplasia is that your immune system is going to target them and kill them. That's what we hope. I don't know how close attention y'all pay to those things, but I was just reading recently about a new technology that is taking up patients' T lymphocytes, the T cells, which are the ones that attack cancer cells normally, and actually stimulating them, training them to attack that patient's own cancer successfully. So they're calling it a new vaccine. It's not really a vaccine, but they're reintroducing the patient's own T cells that they've trained to home in on their own cancer with some success, with some startling success. Cancers that used to be, if they progressed to that point, it was time to go home and put your affairs in order. Yeah, and then there's like no cancer months later, just after this T cell treatment. So it's still in the experimental stages, but you know they're going to be pouring research money into that. That's the most hopeful I've seen cancer researchers be in quite some time. Okay, hypertrophy versus hyperplasia. You see this word root plasia again. Plasia is the same word root as plastic. This really just does not like I's, T's, and L's. Plasia and plastic refer to growth. Plastic means I can mold it, form it. That's basically what it means in terms of manufacturing. In terms of physiology, we use the term plastic to refer to something that can change its shape or continue to grow or whatever. So when you see hyperplasia, then you're talking about growth of new cells. Those cells are regenerating. They're dividing by mitosis and just producing new cells. When you see hypertrophy, that just means the organ itself is getting larger. And in the case of hypertrophy, it could be the individual cells are growing bigger rather than growing new cells. Does everybody see two ways for an organ to get bigger? Make the individual cells bigger or put more cells in there? Hypertrophy is typically growth of the cell. Hyperplasia, increase in the number of cells. Okay, which one are you going to see in skeletal muscle? Hypertrophy, the first one. Why? Why don't you see hyperplasia? Because muscle doesn't regenerate. Muscle cells typically don't divide again. So we typically don't see hyperplasia in muscle. There are some exceptions to that. Smooth muscle, for example, can do it better than skeletal muscle can. We'll worry about those details later. Okay? If I'm looking at a cancerous process, I'd be looking more at which one? Hyperplasia, an increase in the number of cells. Make sense to everybody? Okay, our next term is atrophy. That's kind of the opposite of hypertrophy. So if hypertrophy is getting bigger, what would atrophy be? Getting smaller, shrinkage of tissue, loss of tissue. And we see atrophy in a number of different settings. Typically, you see atrophy of limbs in a person who's been paralyzed. So loss of nerve supply causes an atrophy, not just of the muscle, but also of the connective tissue. Inactivity can cause atrophy of connective tissue. So for patients who are immobile for whatever reason, they may be in a coma, or maybe they're in traction, or maybe they're sedated or whatever. But if you have a patient who's immobilized for a long period of time, what we find is that the ligaments and the tendons and the muscles tend to shorten. So what would happen to someone's hand if you didn't stop it? They'll actually curl into a ball, and we're talking about collagenous fibers, ligaments here. You can't reopen those fingers. The muscles have shortened, the ligaments and tendons have shortened, and you would be breaking bones if you tried to straighten your hands out. So one of the primary activities that are done with those patients is physical therapy to continually keep their hands from doing that, and likewise with limbs. So why do we see someone with end-stage Alzheimer's usually in a fetal position? The same concept. They've been immobilized, and the tendons and ligaments and muscles atrophy, and that just causes them to draw up. Okay? You'll see atrophy in a patient with one side injury, hemiplegia, which is loss of nerve impulses to one side. So if you've ever noticed someone who has a drooping on one side, drooping eyelid, drooping corner of the mouth, sometimes that's viral, that's Bell's palsy, but at other times it is due to stroke or actually damage to the nerve on that side. And what you'll notice if you look at them, and I had a friend who went through a surgical procedure where they had to cut the nerve, and all of the muscles on that side, including the muscles in her neck, all of the muscles were atrophied. So one side was just almost skeletal, and the other side was fully fleshed out. So that's just another example of atrophy. All right, you tell me what you think neoplasia is. We're looking at the word root. We met this word yesterday or maybe last week. Neo is new, plasia is growth, so neoplasia is new growth. And that's the term we use for tumors. Okay, last term on the list here is necrosis. Is that a happy word? No. What is necrosis? Cell death. And it's a cell death in a way that leaves the dead cells in place. So if you've lost the blood supply to a toe, or if you have a bed sore, or you have a venomous insect snake bite kind of a thing, could be a spider bite, you may have necrotic tissue. What does that mean? If it's necrotic, the tissue is dead and it's sitting there dead. What's it doing while it's sitting there? Is it patiently waiting for you to clean it up? No, it's rotting in place. Okay, so nasty, necrotic tissue. In the case of burns, if you have necrotic tissue, what are you going to have to do? Burns typically are superficial, right? So they're going to burn the skin. You have to get it off. It will not heal well if you don't. Okay. So I want to introduce another term that's not on here. Let's see how well I can write it. How do you want to pronounce that? Apoptosis. Does that sound right to you? It doesn't to me, but that's actually right. I don't like it, but that's right. Okay, I'll tell you why I don't like it, and then we'll talk about it, okay? So the word root is ptosis, which is another good word for you to learn. In ptosis, the P is silent. Ptosis means drooping. So if you have renal ptosis, what do you got? Droopy kidneys. The kidneys are retroperitoneal, remember? So they have a fibrous membrane that holds them in place, tucked in with lots of adipose tissue. You lose too much of your adipose tissues, the kidney literally drops along the back body wall. That's a drooping kidney, and that's not a pretty picture because the urine won't flow. Okay? So ptosis, drooping. You may have palpable ptosis. Anybody know what the palpebra is? Well, you get to learn that later this semester. We won't worry about it today. But the palpebra is your eyelid. So if you have droopy eyelids, that's ptosis of the eyelids. Okay? So droopy, droopy, droopy. Apoptosis. Apo means apex, extremity, edge. So in my opinion, and again, nobody ever asked me my opinion when they decide how to call these names, but it ought to be pronounced apoptosis, shouldn't it? Just say yeah. Yeah, absolutely. Because that P ought to be silent, because it's part of the word ptosis. Yeah, no, it's apoptosis, and there's nothing I can do about it. That's just what it is. Okay, well, let's talk about what apoptosis is, and you'll hear me call it apoptosis because, damn it. Apoptosis or apoptosis is programmed cell death. And in the case of programmed cell death, unlike messy, nasty, rotting necrosis, the cell not just dies quietly, it packs up its little parts and disappears. And it does it by a very particular set of steps, which we'll learn more about when we study the immune system next semester, because your immune cells can tell cells, go kill yourself quietly and don't make a lot of fuss about it. And that's what apoptosis is supposed to do. And these cells will then very quietly go shred all their DNA to make sure they don't become cancerous. They have a little shredder machine in the nucleus. They shred their chromosomes. They shut down their mitochondria with mitochondrial poisons. They shut down their metabolism. And then their membranes start to just pinch off in little blebs or bubbles that surround all those internal cell parts so that they don't escape and trigger inflammation. And then neighboring cells called macrophages just eat all the little bubbly parts. So it's like, I'm going to shred my DNA and poison my mitochondria, and then I'll package myself up in all these little bubbles that make it convenient for the macrophages to come in. And that's what apoptosis is. And you do this naturally all the time. Did you know that during the embryonic stage, you had webbing between your fingers and toes? What happened to it? This did. Those cells were programmed to live a certain period of time and then kill themselves. Used to have a tail, went away. But additionally, cells that are abnormal, you have precancerous cells that pop up hither and thither, and your immune system will simply recognize most of them, the majority of them, and tell them to go commit suicide, and they do. Okay, so which one do you prefer, apoptosis or necrosis? Which one sounds better to you? Yeah, let's go with the apoptosis. It's a much nicer way to go. Okay? Have you heard of necrotizing fasciitis? What do you think is happening in necrotizing fasciitis? You have rotting fascias, whatever a fascia is. A fascia is a sheet of connective tissue under the skin. It's the hypodermis of the skin. And it helps to connect your skin to the underlying muscles. And if you get a wound that enters through the skin, you may get bacterial infections that can cause necrotizing fasciitis. And what do you suppose the treatment for that is? Well, the skin itself is not harmed, right? It's just the fascia under it. How are you going to get to that fascia? You're going to have to find a point of entry, usually near the point where the infection was. Make an incision and just start with some scraping device and get in there and scrape all that. Does that sound like fun to you? Yeah. Okay, so some other time, ask me about the grossest story of necrotizing fasciitis that I know. Not now. Maybe after we finish the skin. Okay. We classify our cancers based partly on tissue type and partly on cell type. And some of these will come back to the cancers of the skin we're going to talk about later. But I think you need to know the basic terminology. A carcinoma is a cancer of the epithelium. Sarcoma is a cancer of connective tissue. If it's specific to bone tissue, it's an osteosarcoma. And I didn't list two of the less common ones, which would be a neuroma and a myoma. Why not? Why aren't they common?
Because they're less and most likely
They don't regenerate. Neuromas are tumors of the nerve tissue. Would they be more common in children or adults? Children, because children are more likely to still have some of the immature nerve cells. So those are very, very rare. This always takes longer than I think it should, but we're done. Is everybody good with that? We've still got to get through the subject we were supposed to be doing today, which is the integumentary system. Okay, we're moving into a new era in the course. This is our first system. Everything we've done till now has been background information. So congratulations. We're going to spend the rest of the year on one system after another. And this is the first system, is the simplest. That's pretty much why we start with it. But it is nice as we start a new system to revisit our themes. And the first one is our levels of organization. So we'll be looking at the chemicals that we associate with skin, the cells, the tissues, and the structures of each. We'll be looking at complementarity of structure and function. What did that mean? When they complement each other? It means if you know the one, you can predict the other. Form and function go together. And then we'll be looking at homeostasis of the skin, at least in a couple of ways. We'll be looking at thermoregulation, and we'll be looking at sensory input. Okay? So those are the main themes. We'll come back to each of these in detail later, so don't feel like you need to copy them all down right at this second. You have a great figure of skin in your book. You have in your chapter summary, you have some activities that will help reinforce the terminology associated with it. You also have in your online lab that thankfully is now finally available to you, a section on the tissues and structures of the skin. So do that in conjunction with this test, okay? That integration lab is in your lab canvas. It's, which one was it? Exercise 12. Okay, so get started on that. I'm writing it as we go, but the part you need for this week is written and posted. So we'll come back to that. We're going to look first at the layers in turn and look at the structures associated with each layer. Of course, the uppermost layer is the epidermis. You have a couple of different concepts you need to put in different categories for this. One is what are the cell types of the epidermis, and the other is what are the layers of the epidermis. And the layers of the epidermis, well, that's a concept we haven't talked about before. This is a stratified squamous epithelium, but it is different from all the other stratified squamous epithelia in the body. It's the only one on the surface. And the term we use for it is that it is a keratinized stratified squamous epithelium, and all the internal surfaces we call non-keratinized or moist. This is a dry membrane. The outer surface of it is dry. So you have that diagram in your book, and we'll come back and talk about those parts. Another concept that we need to introduce is the difference between thick skin and thin skin. And this difference is based on location, but also on histology. Thick and thin skin are clearly different from each other. Duh, thick skin is thick and thin skin is thin. And if you look on the left, you see just how thick that thick skin is. And I want you to notice in this picture that they're not even drawn to the same scale. So this part here is the same thickness as this part here in thin skin. So the picture on the right is already magnified more, which means the difference is more pronounced than it even shows in the picture. So is thick skin really thicker than thin skin? Absolutely it is. Is that the only difference? Well, we also find that in thick skin there's an extra layer that we don't see in thin skin. And if we go back to our diagram of the layers, you'll see it here. It's seen only in thick skin. And then the third thing that differentiates thick skin from thin skin is where it's found. you find thick skin only on the palmar and plantar surfaces. So where are we? Where are your palmar surfaces? Palm of the hand, but also the fingers on the ventral side. And likewise, plantar, where's that? That's the sole of the foot and the corresponding parts of the toes. Does that make sense to everybody? Palmar and plantar, that's the only parts of the body that are truly thick skin. Now, that doesn't mean the remaining skin on your body is all the same thickness. You know that the skin on the back of your elbow is thicker than the skin on the front of your elbow, right? Eyelids, thinnest skin. Okay, so not all thin skin is equally the same thickness, but it's still all classified as thin skin. Even though it's thicker here than it is here, this is still thin skin. Why is that? 3 reasons. Location, extra layer is missing. It's not as thick skin. All right, so those are differences in the epidermis, different parts of the epidermis. All right, so now let's focus on these layers and cells. The layers are listed for you, and you can see them in order. Let's see how I've gotten to where I am. Each layer is called a stratum. And, you know, I could go over them with you. I've gone over them in the lab canvas. There's a good explanation of them in that online lab. So I don't want to take class time to go over something that is already available to you. I am going to tell you there are a number of test questions that have to do with, do you know the characteristics of these layers? Can you put them in order? Can you identify them from a picture? Of equal concern to me are the cells. And there are 4 cell types found in the skin. Now, here's something else that's new. We haven't done this before, even though we've been studying epithelium. Is this a stratified squamous epithelium? Absolutely it is. But it's differentiated into these layers, first of all. And second of all, we have a name for these cells. We call them keratinocytes. And again, that's another one that I don't want to pronounce it that way. How do you want to pronounce something that's built on the word keratin? To me, that's a keratinocyte. But I listened to a video from some people who work with this, and I'm sorry they call them keratinocytes. I can't fix it. So we'll have to deal with that. What do keratinocytes produce? Keratin, not keratin. Okay, here's the other cell type that you ought to know. What do you want to call that one? It produces melanin. Melanocyte. It really is a melanocyte, not a melanocyte. Cannot help with that. Okay, so keratinocytes are the stratified squamous epithelial cells. And they're found in these layers. So the cells that make up the epithelium are the keratinocytes. Are you with me on that basic idea? As they move from the deepest layers to the more superficial layers, they produce the keratin. When they get to about here, does everybody see where I circled? When they get to the top of the stratum granulosum, the dark layer, they dump their keratin and they dump it under themselves. Okay, so let me try to draw a picture on the board. So here sits a keratinocyte. More cells are pushing it up as they grow at the base. This cell is producing its little keratin in the form of granules. Remember, a granule is nothing but a little secretory vesicle that's going to dump its contents. When it's ready, it dumps its contents this way. So all these little granules will migrate to the lower border and dump the keratin that it has produced. Not all of it, some of it will just stay inside of the cell. So this little, and remember keratin is waterproofing, water resisting, so it's hydrophobic. So it's like putting down a layer of wax. And where is the blood supply? There's the surface there at the top, blood supply down here. What did that cell just do to its connection to the extracellular fluid, bringing nutrients in? It just sealed itself off. It laid down a coat of wax between itself and its own blood supply. Tell me what's going to happen to that cell. It's going to die. But when it dies, it sits in place with its little waxy layer under it, but still full of waxy stuff. So you could think of these cells when they move up into these layers of dead cells, they're like little sandbags. And they just sit there in place. They are connected by desmosomes. Remember what those do. They stitch the cells to each other. But they're dead. So as damage accumulates, and that damage could be ultraviolet radiation or just abrasion or whatever, the desmosomes start to break. And as they get closer and closer to the top, what happens is they just slough off the top. Make sense to everybody? So if you were a bacteria or a virus or a fungal spore, where are you going to land? Only place to land is on the surface, right? Okay, so tell me about the cells that these potential microbes, or microbes that are potentially pathogenic, tell me about the cells they're landing in. They're dead cells. Can you infect a dead cell? It's really not very easy to do. Okay, and those cells are just going to slough off anyway. Okay, so everybody see the beauty of the keratinized stratified squamous epithelium. Okay, let's talk a little bit about the other cell types. Let me see what's on the next slide. I think we'll skip over that. Come back to that. You have two kinds of cells embedded in the deeper layers of the skin, the dendritic cells and the tactile cells. What does tactile mean? Tactile is touch. Your tactile sensation is your sense of touch. So tactile cells respond to some degree of touch, a light touch, fairly light touch. And in this figure, the blue cells are the tactile cells. And in yellow, kind of in the middle of everything there, are the dendritic cells. The other names for these, Langerhans cells and Merkel cells, those are the names of the guys that first described them. And we're trying to move away from those names, first of all, because sometimes those guys describe more than one thing. So Langerhans not only describes cells of the skin, he describes cells of the pancreas. So if I say Langerhans to you, you might be thinking pancreas. So it gets confusing. So we'll call these dendritic cells. That's more commonly used today, but it doesn't hurt to know the other term. Okay, and somewhere I skipped the melanocytes. Where did that go? Yeah, there it is. So the melanocytes, melanocytes, produce melanin. What does melanin do? What does it do for you? Is it protective or harmful? It's protective. What is it protecting you from? UV radiation. Melanin is a protein that is a pigment molecule. Now, this may seem like, so what to you? You know that melanin is a pigment, right? It's the brown or black pigment. There's a different variety of melanin called pheomelanin that is red, okay? So those of you with the red gene, good for you. It's not as protective, but it sure is pretty. Okay. Pheomelanin, it's just slightly different. Most proteins are not pigments. Most proteins are transparent. And it's hard to imagine that because you think a protein as being microscopically small to begin with, so you're not used to thinking of it in terms of can I see it or not because it's microscopic. But when you look at your skin, can you see through your skin for the most part? Can you see blood vessels through your skin? So tell me about skin cells. Can you see through them? Yeah. So, and they're made of a lot of protein. So what they're made of is mostly, at least translucent, if not transparent. You can see through skin, except where you have enough melanin. And melanin protects you from ultraviolet radiation. It blocks UV. And that's a good thing, or not, depending on how much you have and where you live. So melanin protects fair-skinned people from being sunburned and potentially subjected to cancer. But melanin also blocks UV rays from getting to the keratinocytes, which are also doing something else for us. Let's go back to the one that I missed, and that was this one. What else do keratinocytes do for you? They produce vitamin D. Did you already know that? Sun exposure on the skin, at least 15 minutes on your face and arms every day is probably going to be all you need, because you're probably going to get enough of the rest of what you need from the diet. But if you have darkly pigmented skin, you are likely to have a vitamin D deficiency, and you should probably supplement. On the other hand, you don't need to spend a lot of money on sunblock, which you should probably do. Okay? So vitamin D is necessary for what? What do you need vitamin D for? Bones. And we'll study that when we get into the skeletal system, but we're finding vitamin D does a lot of other things as well. And the science is not clear-cut on that, but we're pretty sure vitamin D has lots of other roles. So you probably, if you're not getting 15 minutes a day sun exposure on your arms and your face with fair skin, more than that if you have darker pigmentation, then you're probably not getting enough vitamin D. And if you're of a certain age, not looking at anybody in particular, if you're at least 30, you should probably be taking a supplement. That's just personal advice. Check with your own physician. Okay. UV ray is harmful in more than one way. UV rays can break down folate. What do we know about folate? It's also called folic acid. It's a B vitamin. Didn't we talk about that yesterday? What did we say about it just yesterday? It is involved, or it is correlated, a deficiency of it is correlated with spina bifida. And not just spina bifida, but other metabolic problems as well. So what happens if you get too much UV? You get a folic acid deficiency. UV breaks down folic acid, okay? So if you get too much UV, you're worried about skin cancer, right? That's not going to be what's going to affect your ability to pass on your genes to others. It's the spina bifida problem. If you're going to get skin cancer, how old are you likely to be when you get skin cancer? Anybody know? A fatal form of skin cancer? Most likely in middle age, after you've already reproduced. So is the likelihood of skin cancer correlated with poor reproductive strategy? No, it's folic acid that's correlated with poor reproduction, okay? So too much UV, yes, we worry about skin cancer. But to our ancestral populations, the problem was not skin cancer. It was the breakdown of folic acid. Okay, so what are the signature chemicals associated with the skin? What two chemicals did we just talk about? Keratin and melanin. All right, moving into the dermis. The dermis has two layers. They're both connective tissues. The upper layer is called the papillary layer. It's made of structures called dermal papillae. So looking at this picture, do you see these little lumps over here? Those little bumps are papillae. A papilla is a protrusion, a bump. And where you see papillae, what you are seeing is folding. And what do we know about folding? Increases the surface area. So if that is the dermis, what does the epidermis do right over it? Doesn't it fold to match, as you can see here? So the epidermal ridges fit down into the dermal papillae. How easy is it to pull those two layers apart from each other? Yeah, it's not going to happen, right? Because it doesn't just peel flat off. It's not a flat layer at all. It's very tightly connected. So that is the papillary layer. It's the layer just that has the dermal papillae, just this upper part. And it is a kind of connective tissue that you should know. You should all be able to tell me what kind of connective tissue that is because it's up under the epithelium. What kind of connective tissue is always up under the epithelium, everywhere? What are your choices? It's either loose or dense. Which one is always under epithelium everywhere? Loose. Okay? So if you didn't know that, you do now, right? Okay, so we'll talk more about that. So how does the surface of the dermal papillae qualify as an example of complementarity of structure and function? The way those layers fit together. When you see that high surface area, what do you know? Something's going on, right? And because they're so tightly connected to each other, you know that shearing forces will not pull them apart. Okay? There's some other things, some other aspects of that we're going to talk about as we get into the blood supply. So this is beyond, a little bit beyond what your book has. So I'm going to point out where the blood vessels are. So we have some that are up there associated with the dermal papillae. We also have some that are way down deep, down at the very base of the dermis. And then we have some capillary loops inside each dermal papilla. And then we also have some capillary loops associated with our things like our sweat glands. Does it make sense that sweat glands need a blood supply? Hair follicles need a blood supply. Okay, so every little structure within the dermis has its own little capillary loop. The dermal papilla, remember, is supplying blood to the epidermis because the epidermis is avascular, right? So when you look at each dermal papilla, what you're seeing is a little fold, right? A papilla is going to look something like that. And what about the epidermal cells associated with it? They sit over the top of it. And then where are these blood vessels? There's just a little capillary whorl in there. And so I can get diffusion of nutrients to my epithelial cells from all sides. So that folding that increased that surface area wasn't just to hold the two layers together, it also increases the area for absorption. Make sense? Okay, so that's a little bit on the blood supply. Let's get some terms. So we have two layers of arteries and veins. The superficial one is up near the papillae, so it's called the papillary plexus or superficial plexus. And that's the blood supply to the epidermis. And these terms are not in your book, or at least I don't think they are. And the deep layer, the one that's way down at the base of the dermis, that's called the cutaneous plexus or the deep plexus. Plexus just means network. It could be a network of nerves, but in this case it's a network of blood vessels. And that supplies blood to the entire skin. It's also the one that can vasoconstrict and vasodilate. Does everybody know what those terms mean? If a blood vessel vasoconstricts, what's it doing? It's squeezing. How much blood flows through a vasoconstricted vessel compared to a vasodilated? Constriction reduces flow of blood. So it's pretty warm in here today. Would you agree with me on that? Yeah. So what about the blood vessels in the cutaneous plexus of your face? What are they doing right now? For the most part, they're dilating. You're trying to radiate off some heat. Any questions about that? Okay, so related to the idea of skin color, or excuse me, of circulation, is the idea of skin color. Now, if you're going into healthcare, you may need to be able to do patient assessment, right? So when you assess a patient, the first thing you do is you notice how they look and how they act. So if you're examining a patient while you're asking them questions, you're looking at their skin, aren't you? For the most part, you're not like propping their eyelids open and examining their eyes first thing. You might do it eventually, but you're not looking at asking them to say, you're looking at their skin. So it's important to be able to assess what you can by looking at a patient's skin. And here are some terms that I think it's important for you to know. Erythema, cyanosis, and pallor. So erythema means reddening of the skin. If you're flushed, whether it's from embarrassment or you have a fever or you just got hot, doesn't matter, it's erythema. Erythema is reddening of the skin, and it is associated with the vasodilation of those cutaneous vessels. Everybody good with that one? There are some particular terms. If it's a rash, if it's a pinpoint rash, if it's an abrasion, we're not even going to get into all that. We'll just stick to the basics. Cyanosis means you turned blue. And turning blue is what occurs when the blood is flowing, but it doesn't have enough oxygen in it. So generally, people are cyanotic if they can't get air in and out of the lungs. Not necessarily the only way to do it, but that's the primary way to do it. Quit breathing. And pallor is just being pallid or pale, turning white. So you may someone see just turn white from shock. It could be emotional shock. Have you ever seen somebody that the blood just drained out of their face when they heard bad news? That's an example of pallor. But it could be that they're hemorrhaging excessively and the blood is draining out. Gunshot wound to the lungs will cause that. Why? How many blood vessels in your lungs? A lot. So if you have damage to the lungs and you're bleeding internally, you got this nice big thoracic cavity, blood's pouring into it, yeah, that's going to cause a lot of hemorrhage. Okay? There are other abnormal skin colors. There are other causes for different colors. I love talking about it. We just don't have a lot of time to do it. But I do want to tell you one quick story of a guy who went to the emergency room. It was like the week after Christmas, and he was off work, and he was concerned, but now is when he had time to go because he was off work. And his skin had turned blue, and he didn't know why. So he went to the emergency room and just said, I don't even know if I need to be here or not. I don't feel sick. I got no symptoms. I can't nail down anything that would cause this, but I've just turned blue, and I just have no idea what's going on. So they examined him. They took his vitals, started a patient record, and then they discharged him because they didn't know what to do with him either. They said, come back if you start feeling sick. We'll keep your record open. Yeah, he called them a few hours later. He'd figured it out. He got brand new navy blue towels for Christmas and had not pre-washed them the way they said you're supposed to before you used them. So you just never know. Okay, we're going to focus our attention now on the reticular layer. This is the deeper layer of the dermis. And we just learned yesterday when we were learning our tissue locations that this is an irregular, dense connective tissue, mostly collagen with some elastin mixed in. So when you get scar tissue in the skin, what did we say about how much elastin you get? Not a lot. We just learned today with tissue repair, you tend to reinforce the collagen because once you've damaged it once, you indicate it needs to be tougher. So those collagen fibers within the dermis of the skin follow patterns. And to prove this to yourselves, look at the ventral surface of your fingers. Do you see not the flexure lines where your knuckles are, but the vertical lines that run parallel to your fingers? Does everybody see those? I'm not hallucinating, am I? Yeah, I see them. Those are cleavage lines, or lines of cleavage, and they're based on the predominant direction that collagen fibers run. We said they're irregular, they run every which way, but they tend to run in a predominant direction depending on where in the body you are, and we can actually map that. Where did my map of cleavage fibers go? There it is. So if you're ever going to have surgery, you want to quiz your surgeon on the lines of cleavage because, as you can see in the middle pictures at the top, if you cut across those, you're cutting across the collagen fibers, and we know collagen doesn't heal very well. If you cut along the direction of the cleavage line, you're cutting in between most of the fibers, and it tends to heal up more nicely. So whether you get a gaping wound or a nice clean incision that heals well depends on the direction of the cut. Did you know that? Do you have any injuries that you can look at and go, that one, yeah, that's right across. Usually when I cut my fingers, I've got a couple of them. Here's one that's still there. I'm cutting frozen bagels or onions or something. If the knife slips, I'm cutting across, not along the lines of cleavage. Yeah, there's some pretty ugly scars there. A couple of them. Cut the tip of my thumb off once. We don't need to talk about all that. Okay, so that's the importance of the cleavage lines. Okay, so next, skin functions. I am assigning you to read your book about the functions of skin. And I want you to notice when you do that I've kind of lumped a lot of things under protection, because I think that the book is a little too simplistic in saying one thing's protection, the other one's not. So when you think of protection, the skin's protecting you. What are you being protected from? Any possible insult in the external environment, whether it's ultraviolet radiation or microbes that want to infect you or dirt that wants to get in and, whatever. So you've got an ultraviolet barrier, you've got a physical barrier, you have a barrier against water loss or gain. You have your immune cells, which includes those dendritic cells. You protect yourself from overheating. You protect yourself from too much heat loss. And certainly your sensory input is a protective mechanism as well. So I would argue that all of those are protective, even though your book only lists one of them, or maybe two at most, as being protection. And then you have a couple of metabolic functions. You produce vitamin D in your keratinocytes. That's an absolutely important step that we'll look at in more detail when we get into the bones, which is where they're going to do their thing. And then there's some organics that are secreted. The secretion of these organics is kind of peculiar. And it's not just organics. They're the ones that are metabolically secreted. We already talked about sweat glands secreting salt and water, right? But there are also waste products like urea that are included in that. And it's not because you need a way to secrete urea. Your kidneys do a bang-up job of getting rid of all the urea you need to get rid of. So that urea must be there for another function. It is osmotically active. It pulls water. So that's probably part of it. But in certain sweat glands, the ones we associate with the armpits and the pubic region, those sweat glands secrete organic molecules that can be attacked by bacteria and broken down and create the characteristic odors that we spend big bucks trying to cover up or stop the secretion of. So all of those organics have a function. We don't necessarily understand the function of all of them, but I will tell you that pheromones are documented in other animals and at least partially confirmed in humans. So what's a pheromone? Anybody know what a pheromone is? Anybody? You know what a hormone is, right? A hormone is a chemical secreted by one tissue to affect the action of another tissue somewhere distant in the body. A pheromone is a chemical secreted by one individual that gets out into the environment and impacts the behavior or physiology of another individual. So the pheromones we're most familiar with are the ones that are involved in mating behavior of different animals. We know insect pheromones. We've got them down to where we can use them as bait to attract pest insects. So we can bait a trap with pheromones of a particular moth, and it attracts all the male moths, and then we can kill them all so that they don't go and infest our crops. We also know that pheromones are found in vertebrates. How does every dog in the neighborhood know when that one dog is in heat? How do you think they know? Okay, so pheromones are well documented. Evidence of pheromones in humans is interesting. You have in your nasal passages. Right in the very front, you have what's called the vomeronasal organ. Vomeronasal. The vomer is the bone, and of course that's the nasal cavity. Vomeronasal organs are well documented in other animals. If you've ever gone to the zoo and watched the mating behavior of the deer or seen it on the nature documentaries, what do? Anybody want to talk about that? They tend to urinate a lot, but they also are very interested in the females, urine, feces, tail ends, whatever. They tend to curl their lips a lot, and as they curl their lips, what they're doing is they're bringing their lips closer to their nose, and they may use their tongue as well. Okay, and snakes, we know, sample their environment with their tongues, and then they'll stick the tips of their tongues up in this little vomeronasal organ, and it's a way of sensing the environment. So the question is, do humans have a functional vomeronasal organ? And if they do, how does it work and where the heck is it? And so in some people it appears to be non-functional, but in other people it does appear that it is functional. And unfortunately, or whatever, for whatever reason, most of these studies are done by the perfume industry. Why would that be? Yeah, they got big bucks involved in selling scents. So if you're familiar with using musk as a scent, we usually sell that in men's colognes, right? What's that supposed to do? Yeah. It's supposed to make you really sexy to the ladies, I'm just saying. So how does it work? Have you ever smelled musk? I hate to say it smells musky because that doesn't really help you, but that's where that word comes from, and it's hard to describe how it smells. But the thing is, I don't know what it is, but I've talked to people who said, yeah, I really have a reaction to that, and other people were like, I don't know what she's talking about. That doesn't do anything for me. So I think it's extremely individual, but I think it helps you to know where the musk is collected from. Musk is a secretion from the musk glands of male vertebrates of some kind or another. Musk ox, obviously. Musk rats, yes. But other animals as well. But the musk glands are part of the anal glands of the animal. And so when you're using musk, guys, just remember that that's what you're doing. Any questions? Okay. If you have questions that you want to ask about that, feel free to write it on your paper. And I promise that I will anonymously read any questions I find that are entertaining or enlightening at the beginning of class tomorrow, and we'll address any issues you may have. Okay, we are almost out of time. I'm very comfortable with finishing tomorrow, rather than trying to rush to finish today. We are going to go very briefly through the epidermal derivatives, which is the glands, the nerves, the, what else was it? Sensory nerve endings and other structures. I think you'll be fresher if we do that tomorrow, but we still have 2 minutes. So answer the first of these 2 questions. How does the skin serve the body's regulation of heat? Just in 1 or 2 sentences. Give me a simple answer to that. And then we'll work backwards. And we'll finish up tomorrow.